And introducing Dr. Gregory Ordway. Dr. Ordway received his bachelor's in pharmacy from Ohio State University and his PhD in pharmacology at Ohio State University. He was a neuropsychopharmacology trainee at University of Pennsylvania School of Medicine, and he received his postdoctoral fellowship in the Department of Psychiatry at Pennsylvania School of Medicine. He is the senior advisor to the Dean for Research at the James H. Quillen College of Medicine. Please join me in welcoming Dr. Ordway.
Thank you. It's a pleasure to be here, and thanks for coming. Let's see where the little button is. I have to tell you that I have a conflict of interest today. So I'm a co-inventor, along with Adela Zebani and Russ Brown, on a patent application that's been submitted for the use of PARP inhibitors for the treatment of depressive disorders. And I also have a commercial relationship with Drug Shop LLC, which is a startup in Houston. And we have the goal of synthesizing and developing and licensing compounds based on this discovery. So that said, obviously I'm going to be talking about experimental findings and the experimental use of drugs, but at this point we're talking about animal and cell culture studies, and we're not anywhere near to the point of talking about treating humans. But nevertheless, we've got some really exciting findings, and I'll start by giving you a little background of how we came about and how we became interested in PARP inhibitors as potential antidepressant drugs. And I'm speaking to a psychiatry audience, so I don't have to tell you how prevalent major depressive disorder is. And this is an estimate of the economic impact of major depression, but it's probably on the low side, because major depression, if you have it, it increases your risk significantly for the development of a variety of different medical disorders, not to mention increased risk for mortality. So it's a complex illness that's interwoven into many different medical and psychiatric disorders. The use of antidepressants, therefore, is widespread. And somewhere around 1 in 8 individuals takes an antidepressant drug in the US. So in an audience of, say, 30 here, there's at least 3 or 4 people that are probably taking an antidepressant now. And that comes to a total, it's hard to believe, 250 million prescriptions written per year for antidepressant drugs in the US alone. So that's a lot of prescriptions, and it's not being written by, most of those are not being written by psychiatrists. Most of those are being written by family medicine doctors, and et cetera. So if you can't read this cartoon, it says, of course you feel great. These things are loaded with antidepressants. So this is just a quick look at the timeline of development of antidepressant drugs, and I will tell you that the development of antidepressant drugs is characterized by very long periods of low level of innovation. And I'm not sure if you could even use the word innovation anywhere in the history of the development of antidepressant drugs. So if we go back to melancholia and early treatment there with bloodletting, the big advance was the use of leeches, where you didn't have to cut the person. And then somewhere before the 1900s, we had sedatives being used widely for many different psychiatric disorders. And the modern pharmacological age of treatment of depression was in the mid-50s with the accidental discovery of 2 drugs with antidepressant activity, MAO inhibitor and a mixed norepinephrine serotonin uptake inhibitor. Those were accidental discoveries while searching for drugs with other activity. And then based on that discovery, and I'll say that these drugs happen to increase norepinephrine and serotonin in the brain. It wasn't that somebody found that there was an imbalance of norepinephrine and serotonin or low levels of those transmitters, and therefore predicted that drugs that would elevate those transmitters would have antidepressant activity. Rather, they accidentally discovered these drugs and later discovered that these drugs elevate norepinephrine and serotonin, and hence was born the monoamine hypothesis of depression, that depression is some imbalance of those neurotransmitters, and it's based primarily on the pharmacological actions of those accidentally discovered drugs. So through the last 60 years then, when I talk about low levels of innovation, this is really where I'm talking about. Probably the major advance here was the development of fluoxetine in the 80s. Actually, it preceded that, but it was released in the 80s. And it was a novel finding or a novel development in the fact that they were able to produce an SSRI, a serotonin uptake inhibitor, that did not have the toxic effects of the older drugs that were discovered accidentally. So that was the innovation of the last 60 years, was the discovery of a drug that didn't have some of the toxicity, still had side effects, but it was an effective antidepressant without the toxicity. And then from that came many more antidepressant drugs to today, where we have 40 slightly different antidepressants available worldwide for the treatment of depression. Most of those drugs are inhibitors of serotonin uptake or norepinephrine uptake or some combination of those things, or a few monoamine oxidase inhibitors. And they're all based on tweaking the molecules, basically. So I know that when the drug companies tweak these molecules, they say, ooh, look at this, we added this little tidbit of serotonin antagonism or something like that. But this, to me, is very low levels of innovation, still based on a very old accidental discovery. So the question is, okay, the antidepressants, they are very effective in people. Why would you bother to search for more? Well, they're very effective in some people, but unfortunately, a lot of people, they're not very effective. So if you look at this antidepressants response rates, depending on who you read, is around 50 to 60% of depressed patients, and response rate is judged by a 50% reduction in a HAM-D scale. So that doesn't mean they're remitted. It just means that they feel better, but they're still not back to normal. So remission, true treatment, effective treatment, occurs in less than 50% of depressed patients. And approximately 30% of depressed patients will not remit after multiple trials of antidepressant drugs. And according to my calculations, that leaves about 2.4 million people in the United States alone that are untreatable or poorly treatable with existing antidepressant drugs. And that's despite vast sums of money that have been dedicated to research and development on depression and its treatment. I will mention again that this money that's been put into the development, this is public money and private money, is largely centered on, I don't know what percentage, I'd like to find that out, what percentage of that money has been dedicated to the development of more monoamine drugs, with a very small part of that money being looking outside the box. So my lab, for my career, has been focused on trying to understand the biology of depression, and we do that by studying brains from people who had depression prior to death and comparing the biochemistry in those people to the biochemistry of people who are psychiatrically normal prior to a natural death. And our guiding principle is that if we can find pathology, we should be able to identify new targets for the treatment of depression. I would say the first 20 years of my 30 years of doing this business was devoted primarily to studying norepinephrine, serotonin, and dopamine systems in depression, with the idea that maybe we'll, based on the pharmacology of these drugs, we'll find something wrong here, we'll find some new targets. That was guided by the NIH's funding approach. I mean, years ago, if you wanted to get funding for research on depression and you weren't studying norepinephrine and serotonin, you could pretty much forget it. So 20 years of my research was dedicated to studying primarily norepinephrine. We found some very interesting things, but never found any new targets that we might be able to use to develop a new antidepressant drugs. And about 10 years ago, we started shifting our focus away from norepinephrine and serotonin, and more intensely in the last 5 years, we've come far and away from norepinephrine and serotonin. I will say at the same time, it's becoming, along with other factors, extremely difficult to get funding from the NIH when you don't have norepinephrine and serotonin and you're not talking about that. So we've become very interested in the role of oxidative stress and oxidative damage in depression. And of course, it's well known that oxidative stress is involved in a lot of disorders, medical, psychiatric. But some of the features of oxidative stress findings in major depression are unique, and the data are quite strong, demonstrating that there's oxidative damage to tissues in people who have major depression. This is just a list of a bunch of findings from various investigators showing either elevated levels of oxidative molecules, oxidative damage, or low levels of antioxidant molecules or antioxidant enzymes primarily in blood tissues from people who had major depression compared to normal control subjects. And then some of this data has been recapitulated in animal models of depression. So there's a vast literature showing elevated oxidative damage, and this is intertwined with the literature on the role of inflammation in depression, because inflammation elevates oxidative damage and creates oxidative free radicals and oxidative damage. So one of the pieces of this evidence that I'll draw your attention to that got us really going was a finding by Naomi Simon at Harvard, who showed shortened telomeres in telomere DNA in blood from major depressives and normal controls. And her data was brought about by previous investigators showing that prolonged psychological stress in humans resulted in shortened telomeres. This data is not from Naomi's work, but another study that verified it in a much larger population of depressed subjects. So this is really kind of even though hers was a pivotal study, this one really kind of nailed it. And in this study, with large sample size and looking at telomere links in leukocytes from major depressives, they showed, again, a reduction in major depression that correlated with the severity of the illness. A very strong demonstration of, okay, if I can get rid of that, of telomere shortening in major depressive disorder. So let me just remind you quickly about telomeres. Telomeres are non-coding repeat DNA structures that cap the chromosomes at their ends and prevent recombination events and protect the chromosomal DNA. And telomeres shorten with age naturally, and with each division of the cell, they get shorter. So that's a natural event. So those studies obviously are age-matched subjects. So naturally your telomeres will get shorter, but in depression you have much shorter relative to somebody who's not depressed at your same age. But so what's thought to be happening is that telomeres are very uniquely susceptible to oxidative damage, and that's contributing to the shortening of telomeres. So this is sort of our entry into the oxidative stress effects in depression, as we're really excited or intrigued by the shortening of telomeres. It seemed like such a strong cellular effect that's related to severity of illness. So all those studies were done in blood cells with telomeres, and a couple people had done some work in brain, but they just ground up cerebellum and measured telomeres in depressed subjects and normal subjects, and they found nothing and concluded, well, this is just a peripheral thing, not a brain thing. But we knew that that brain cells are not equally susceptible to oxidative stress. There are some cells that are very sensitive, and two cell types that are very sensitive are hippocampal CA1 pyramidal neurons, largely known from ischemia research, and then oligodendrocytes are probably the most sensitive to oxidative stress in the brain. Of course, oligodendrocytes provide the myelin sheaths and create this, nodes of Ranvier that accelerate the transmission of impulses down axons. So these cells are very highly active metabolically because they're constantly producing myelin. And this is a very dynamic process. It's not like you get myelinated and you're done. You're constantly changing the myelination throughout life. And these cells, because of their makeup, they have very high levels of a pro-oxidant iron, and they have low levels of antioxidant enzymes for whatever reason. So we looked at, I'm a part of a group that has a large brain collection of human brains from psychiatrically diagnosed subjects, post-mortem, and we have psychiatrically confirmed normal control subjects that died of natural or accidental causes. And we looked at a group of major depressives and control subjects and doing the usual matching for all kinds of different things. And we excluded psychotherapeutic drugs. If it was in their toxicology, they weren't included. And we excluded antidepressant treatments, even though the data suggests that antidepressant treatments do not alter telomere shortening. So the prominent causes of death were heart disease and the controls, and cardiovascular disease is also associated with shortening of telomeres and oxidative stress. So that's kind of working against us. And then our depressive group, primarily these people died as a result of suicide. And as I mentioned, they're carefully age-matched subjects. And we measured relative telomere lengths here on the y-axis in 11 to 12 normal controls and matched major depressive subjects. And we captured oligodendrocytes using a very sophisticated instrument that allows us to take a tissue section through a brain block and then identify specific cells we label with an antibody, in this case the antibody CMPase that selectively labels oligodendrocytes. We capture those cells and we can isolate the DNA and measure telomere length. We measured telomeres initially in this paper here in two brain areas, frontal cortical white matter and uncinate fasciculus, which is down in the amygdala carrying emotional information. And we found highly significant reductions in telomere length, suggesting that the telomere shortening does occur in major depression in the brain as well. It's just isolated to specific cell types. And then after finding that and getting some criticisms from grant reviewers, we thought, well, because people don't want to think about white matter and glial cells as having some significance to psychiatric disease. So they wanted to know, well, is it isolated to limbic brain areas? And we were kind of hoping, yeah, it will be. And we looked in occipital cortex, white matter, and we just pulled oligodendrocytes out of a gray matter area down in the brainstem. And we saw the reduction in telomere length no matter where we looked. Maybe it's a little more robust here. I don't know. These are done at different times. It's clearly significantly shortened telomeres, pretty much in I would guess, any oligodendrocyte you pull out of the brain in major depressive. And then we looked at astrocytes from the same brain areas, two of these same brain areas. And in astrocytes, which are kind of revved up antioxidant system, they showed no shortening of telomeres in the same groups of subjects. So we were, again, intrigued by this concept that telomeres are short in major depressive. It occurs in the brain in susceptible cells. What are the mechanisms that result in shortening of telomeres? And might we find some novel targets that we could study for developing antidepressants by studying how telomeres get shortened? And of course, as I mentioned, there's a number of things that control telomere length, including aging and replication. But certainly, DNA oxidation is what everybody predicts is going on. And when DNA gets oxidized, there are a whole host of there's a cellular response to oxidized DNA that corrects that. So this is something I'm going to talk about and focus on today, a couple of years ago I talked about these things, which were very interesting, had interesting findings there, but didn't lead us to anything that we could target as a potential antidepressant drug. So we first looked at DNA oxidation, and we took white matter from the same subjects that we had studied, the telomeres, from major depressive donors and psychiatrically normal controls. And we saw a robust elevation of DNA oxidation in the major depressive subjects compared to the controls, measuring 8-hydroxydeoxyguanine, which is the major oxidative metabolite, or DNA. It's a major nucleotide that's oxidized during DNA oxidation events. So there's a robust elevation of DNA oxidation in white matter of major depressives. So what happens when you have DNA oxidized? Well, it induces a strand break. There's a whole host. This is a very complicated process, and prior to our discovery, I knew nothing about this process, and I'm getting better and better at understanding how this takes place. But when you have a strand break from the oxidation, there's a group of enzymes that get activated that come in and repair the oxidized nucleotide and replace it with the original nucleotide, with a non-oxidized guanine in this case. So it starts with PARP1, which is sort of a detector of oxidized nucleotides. And PARP1 then initiates a cascade of events that brings in other enzymes that form a complex that then go and fix the oxidized DNA. So PARP1 was an interesting enzyme, and I'll tell you why. We got really focused on that. We've looked at others. There's some other interesting things there, potential targets, but for the time being, with limited funds, we've focused on PARP1. So we measured PARP1 gene expression. This was we had a lot of things to look at in the DNA repair pathways, and Adela Zebeni, who was in my lab at the time, he chose PARP1, I didn't. He thought this would be an interesting thing because it gets upregulated during DNA damage. And we measured PARP1 gene expression in oligodendrocytes from the same group of subjects and saw a marked upregulation of the expression of PARP1 gene in oligodendrocytes from major depressives in both brain areas that we were studying, this amygdala white matter area and BA10 white matter. And then we also measured it in astrocytes captured from the same subjects. And where we didn't see a change in telomeres, we did see some upregulation of PARP1 in the astrocytes, comparing depressives to controls. So let me recap this before we move on. Major depression is associated with a variety of indices of elevated oxidative stress. And there's a very large literature on this. And it's thought that this is related to metabolic hyperactivity that's induced by psychological stress. This gets a little fuzzy, the proof for that, but generally speaking, that's how people think about it. And then oxidative stress indices are elevated in depressed subjects in blood cells, but also in sensitive cells in the brain, in particular oligodendrocytes. And in the brain, this oxidative stress damage is evidenced by shortened telomere DNA, elevated DNA oxidation, and elevated DNA repair, in particular elevated expression of the PARP1 gene, which is a DNA repair enzyme. So now I'm going to delve into PARP1, and I don't want to lose you here, and I don't want to get you maybe there's some chemists in here, I'm sure there are, they get excited about this, and other people might doze off. But PARP is a really interesting enzyme, and it uses NAD as a substrate and takes the ADP ribose from that and covalently attaches it to proteins to modify their function. And not only adds the ADP ribose, but it makes a polymer on that protein. And you can see that these ADP riboses are heavily negatively charged. They branch, so that changes the it changes the activity of the protein, usually inhibits if it's an enzyme, or changes its binding activity if it's a transcription factor, because it adds a huge, bulky, negatively charged moiety to the whatever protein that it's adding it to. It also self-parlates. So PARP1 activity is high. When it gets parlated, its activity goes down. So that's how it controls itself. And then once proteins are parlated, and this is a polymer of PAR, they get deparlated by PAR glycohydrolase, or PARG. And that cuts the PAR off from the protein and restores its activity. That's the cycle, and it utilizes NAD, which is an important source for energy for cells. Again, this is a complicated slide, and I'm going to simplify it for you, and I just throw it up here because I want you to know that PARP is activated by DNA damage, but there are other activators and other inhibitors. So it's a very complex and complicated regulation of this enzyme. And once it's activated, parlates other proteins and affects many different cellular functions, including it. So any one of these could be involved in depression or antidepressant action. So I'm leaving it open. We have I can tell you why we're interested, but it's possible that any of this could be playing a role. So when proteins get parlated, you get two in particular that we're interested in are the factors that are parlations that occur or non-parlations that occur that activate NF-kappa B. NF-kappa B is a group of transcription factors that drive inflammation. So NF-kappa B is essentially a transcription factor or a group of transcription factor that activates the transcription of inflammatory mediators like cytokines. So that's immediately when we read this, we didn't read this and then study PARP. We did the PARP and then read this. We got really excited because of the potential role of inflammation and its feedback in generating more reactive oxygen species and its potential role in depression. But also, PARylation changes the structure of the chromatin. It relaxes it so you can have transcription factors come in to bind and change transcription. And of course, it's involved in DNA repair. So it's a very complex enzyme that modifies a lot of different cellular functions, but the two that really caught our attention and got us excited was the inflammation story. So PARP activation enhances inflammation. Another thing that PARP activation, how PARP activation feeds into inflammation is, as I mentioned, there's an enzyme that clips the PAR, over here. There's an enzyme that clips the PAR from PARP and other PAR-related proteins. And when that PAR is released from the proteins, it escapes the nucleus, which is where this is all taking place, and escapes the cell and acts as a damage-associated molecular pattern that stimulates inflammation. And then PAR can also have negative effects on mitochondria and result in cell death if it's at very high levels. So PARP activation can enhance inflammation through NF-kappa B activation, but also through the release of PAR that can activate inflammation. And this is a system that's been studied very heavily in the cancer field. And generally speaking, PARP activation will have different effects on the cell depending on what level of activation it has. So at low levels of activation, it can work in immunodefense and inflammation, but at higher levels, things get worse until it's very high levels, you have cell death. I think we're talking about in depression here, and when I talk about inhibiting PARP and looking at that as a potential antidepressant, we're not talking about knocking out PARP because that would probably have some strange effects, could cause cancer. I'm not sure what it could do. We're talking about knocking down PARP We're not knocking it out. And this is a criticism that I've gotten from people. Well, if you knock out PARP, we're not knocking out PARP. We're treating with a drug that's on board for some period of time. It's not canceling PARP. So we devised this general hypothesis to think about how to go about studying this. And so we have the generation of reactive oxygen species, either intracellular or extracellular because of inflammatory cells. This occurs as a result of psychological stress. We know that. We don't know exactly the mechanisms of how that occurs, but it occurs. And the elevated reactive oxygen species then attack DNA. Guanines on DNA are very susceptible, which are rich in telomere regions. And that stimulates the DNA damage repair pathways. And in that pathway is, in the early stages of that process, is PARP1 that parlates proteins, changes their activity, recruits them in for the DNA damage repair. But PARP1, when it's activated, it also has these other actions. It activates NF-kappa B, that's pro-inflammatory. When it's deparlated, PAR can have pro-inflammatory effects. PARP competes for NAD, its substrate, with all the other processes in the cell, including the citric acid cycle. And high levels of PARP can reduce NAD levels and deplete ATP. And also PARP activation, as I mentioned, can cause chromatin remodeling. We predict that this contributes to compromised cellular function, which contributes to the behaviors of depression. That's our general hypothesis. So that brings us to PARP inhibitors. So again, this is, I'm just telling you how we've came to this point. So we found this, we didn't know much about it. Then we started looking at, well, what if we inhibited PARP? What would that do? And PARP inhibitors are on the market for the treatment of some BRCA-associated cancers. So what happens in cancers, DNA-damaging anti-cancer drugs damage the DNA of cancer cells, and those cancer cells adapt by upregulating PARP to increase the repair. So what pharmaceutical companies have discovered is that if you block that cellular response, then you get better damage from your DNA damaging anti-cancer drugs. So it's an adjuvant therapy. But other people, we discovered this after the fact, have found that PARP inhibitors have neuroprotective and anti-inflammatory effects in a variety of different animal models. And how this works is believed to at least be in part by blocking pro-inflammatory effects of PARP activation, i.e. through NF-kappa B. But it's not 100% known exactly how these drugs show protective properties in these illnesses. So we, based on this general hypothesis, we thought, well, what if we treat animals with a PARP inhibitor in a depression model? Would it produce an antidepressant effect? And Russ Brown and I don't know if he's here today, I guess not. He's my collaborator on this, and we talked about this in my office. I remember it was about 2 years ago, and we thought, well, you know, if we block PARP, we're blocking repair, things could get worse, not better. So we decided we're going to do the experiment anyway. If it gets worse, that's interesting, we'll publish it. If it gets better, wow, that'd be amazing. Again, we're talking about blocking this component. I really don't believe that we're talking about activation of PARP to such a level that it's causing cell death. There's just not enough data in the literature to support that. So PARP inhibitors have interesting properties, and we knew there were a lot of them available, but they all have this motif here, this benzamide, every one of them that's in the pipeline and on the market. And that comes from one of the original drugs discovered to inhibit PARP, 3-aminobenzamide. And you can buy this from Sigma. We didn't have to call a company or anything. So we chose 3-aminobenzamide and another 5-AIQ, also commercially available, also containing the benzamide structure. So all the existing PARP inhibitors all have this basic unit to them. And so we started with these drugs. So we had to pick animal models, and animal models of depression are a funky thing, and some people believe in them, some people don't. I would say I don't believe that rats get depressed like humans, but they are good models to discover antidepressant activity in drug molecules. So the first one we actually used a combination. We used a social defeat model plus a chronic unpredictable stress model, and I just thought I'd give you a little idea of, I'm not sure which thing I'm using here. Oh, here it is. This is what so in this social defeat model, you take a retired breeder rat and you house him with a female for about a week. And sometimes we switch the females. But they're used to having a female in their cage. And then you take that big husky breeder rat, or you take the female out of the cage and put a unsuspecting female, smaller rat, male rat, into the cage. And of course, things don't go well for the smaller intruder rat. This is just to illustrate what happens. I'm having trouble with it. I'm sorry. I don't have any way to, there it is. Very hard to see. We'll see if this works. If it doesn't, it's no tragedy. All right, here's the... So the large hooded rat there is the breeder rat who's been housed with a female, and now this unsuspecting male is dropped into the cage, and there's an immediate attack. And then you stop it right then. The animal's not harmed by that. And that's done on a daily basis for 10 days. And we mix that with another chronic unpredictable stressor throughout the day, which is, a mild stressors like tipping the cage or crowding it, exposing them to a little cold water, etc. We did that because rats adapt quickly to stressors, so we wanted a second stressor that was unpredictable that would keep them from adapting. Both these stressors produce anhedonia and social interaction deficits, and we measured anhedonia by measuring their preference for sweetened water. Rats love sweetened water, but if you stress them chronically, they don't care anymore about whether it's sweet or not. This is the paradigm. It's 10 days of stressors. We have handled controls, and we have rats exposed to SDS and CUS, the two stressors, every day. In the last 3 days of the paradigm, we measure their sucrose preference with a two-bottle choice task. And we had 3 treatment groups: vehicle, fluoxetine, which is a SSRI, and 3-aminobenzamide. And then we measured a social interaction on the 11th day, at the same time after treatments. And then we euthanized the animals to measure oxidative DNA damage and PARP1 gene expression. So that's just the model up there, so you can go back to it if you want to look at it. First, to show you, we wanted to know whether the chronic stress in the animals would result in the same biological changes that we see in human major depressives. And in fact, we saw an increase in DNA oxidation in white matter and in prefrontal cortex in a comparable area in the rat. It's not as big as the humans, but this is only 10 days. And we also saw an upregulation of PARP1 gene expression in these stressed rats. In terms of their behavior, they demonstrated exactly what we predicted. They no longer preferred sweetened water over plain water. So they exhibited what is referred to as anhedonia in the animals, which is the phenotype similar to a human major depressive. And they also had reduced interaction in a social interaction test. Then to look at the effects of fluoxetine, now we haven't done this up here yet. We haven't looked at the biological measures. But we did see that fluoxetine, as expected, prevented the anhedonia. But so did, wow, this thing just jumps. Sorry. I don't have a small enough thumb. 3-aminobenzamide also produced an effect on anhedonia just like fluoxetine. And same with the social interaction test. 3-aminobenzamide prevented the reduction in social interaction produced by the stress, just like fluoxetine. In fact, it was actually a little better than fluoxetine. So we were very excited about this data. We thought, wow, I really couldn't believe it. So we thought, well, we better do another model, and we used another model. I won't show you that one, but this is just it's a swim test where you drop the rat in a tank of water, and they will swim for some period of time and then just float. And the floating is considered a sign of helplessness. So they've given up and they float. And oddly, this paradigm is what's used widely in the industry to find antidepressant drugs. For some reason, it's not even a chronic paradigm, it's just a 2-day thing. But it's very useful in finding antidepressant drugs. And there are false positives, and we looked at that potential. So we did the same treatment paradigm, same 3AB. We also did 5-AIQ, a structurally dissimilar but PARP inhibitor. And we did the same treatment paradigm, and then we did fluoxetine in a standard model where you just treat 3 times over 24 hours. We did 3AB that way too. Fluoxetine's been tested in this paradigm so many times that people know exactly the best way to do it, and that's the best way to do it to get the biggest effect. So that's that paradigm, and here's the data. And as expected, immobility was dramatically reduced in rats treated with fluoxetine. That's the standard by which everybody uses to prove that this is a very effective model to pick up antidepressant activity. And we found that 3AB also administered that same way, reduced immobility pretty much to the same extent that fluoxetine did. And 5-AIQ also reduced immobility. We used just 1 dose here. We took it out of the literature. We need to do more work with that drug to find out. We may be able to get further than that. So this data, again, showed that 3AB and 5-AIQ PARP inhibitors have antidepressant activity in this behavioral model, standard behavioral model. So next we were curious that whether if we added 3AB to fluoxetine, would we get an additive effect or maybe synergistic effect? So we treated rats in the in this model, and we treated them with 3AB at a dose that we had previously shown was too low to produce a significant effect. And we chose from the literature a dose of fluoxetine, which was also too low to produce an antidepressant effect, but it has a little effect. And then we combined the 2 drugs in a separate group of animals. And in fact, 3AB, when added to fluoxetine, both at subtherapeutic in rats levels or doses, produced a significant reduction in immobility in the swim test, suggesting that 3AB will have an additive, at least additive, maybe synergistic effect with a standard antidepressant like fluoxetine. This is just to show that the false positives are increased activity produced, like amphetamine is a false positive in the swim test, and there's no effects of 3AB or combinations or anything on activity. So we ruled out the false positive. So let me just summarize. DNA oxidations elevated in white matter in major depressive disorder. We were able to demonstrate with chronic psychological stress that this is recapitulated in rats in brain white matter. DNA oxidation is associated with elevated PARP1 gene expression in major depressive humans, but also in psychologically stressed rodents. And at least in rodents at this time, inhibition of PARP1 with PARP inhibitors produces antidepressant-like effects. It reduces anhedonia and increases social interaction in these models. And we were able to demonstrate that PARP1 inhibition boosts the antidepressant activity of an SSRI when administered at a low dose. So this, coming back to our model, we feel strongly that PARP1 is a really a unique molecular target for the potential development of a novel class of antidepressant drugs. And we're really excited about it. There's a lot of naysayers out there, I can tell you that. We have 3 grants in review right now to fund this research. We're other mechanisms as well. So this is one of our grants is really focused on trying to figure out what's going on with PARP inhibitors and how they might be working. We've also partnered with, as I mentioned in the second slide, with Drug Shop LLC. And this is 2 of our collaborators at Drug Shop. This is Corwin Miller, it's Dr. Mary Miller's son, and Errol Bakabasi. Both of them have this startup company in Houston, and they're very interested in this concept that PARP inhibition may be a novel target for antidepressant activity. That's about all I can tell you about that project. We have an SBIR grant that's been submitted to pursue this to the NIH. So what I want you to be sure you understand is that drug development is a very complex, long process. It's extremely expensive. And this is where we are here. We've got the concept. We've got some structures that we know are effective. We're tinkering with this right here, right now. We're in the very early stages of preclinical development. With Drug Shop, we hope to get this far, mostly designing new molecules and drug development in this preclinical stage, animal testing. And then, from there, we would hope to allure, get major drug companies that have the hundreds of millions of dollars that are necessary to do the clinical trials, et cetera. But that's pretty far down the road. Ultimately, our work is intended to find better treatments for the millions of people that have depression that are not being effectively treated with the current antidepressants. So that's really what's driving our research. It's always driven my research. We are teaming up with industry because we've really had trouble getting funded by the NIH through the traditional approaches. So we've teamed up with industry, and we're hoping through the Small Business Innovation Program at NIH that we'll be able to get funding that way. We're also pursuing grants the usual way, but we're still hopeful that we can finally convince people that this is worth pursuing. So we're Drug Shop, Errol and Corwin and I are very excited that we've kind of stumbled upon each other, and we're hoping that this does not illustrate the direction that we're going. We have much higher hopes than that. And I just want to conclude by acknowledging the many people that have contributed to the project. Ordway was really very important. He's a co-inventor on the patent because he's the one that brought PARP1 to my attention, insisted that we do this. Actually, he also insisted that we do telomere shortening. So I'm not easily convinced of things, but he kept nagging at me about it, and I gave in, and then that really led to the whole saga that I've gone through today. And then other people in the lab, I have students in the lab, Jacob sitting here working on this project, Caroline DeBoard, Wesley Antango, Wei Wang's my postdoc, Michelle Chanley's always involved. Russ Brown's my behavioral pharmacologist collaborator, and his crew is also very much involved. The brains come from from a Center grant, this one. The brain collection's funded by the NIH, even though they're not funding our research on it. This is the grant to ETSU that renovated the building that we have to cite, but it's not funding the research. I'm just pointing that out to you. American Foundation for Suicide Prevention has funded our research, some bits and pieces that are not really particularly related to what I presented today. So by and large, the research that you've seen, most of that's been funded by our College of Medicine. So we're hopeful that we can pay that back in the sale of some licenses on some novel compounds to the university in the future. And with that, I believe that's my last slide. I'll take any questions. Go back to that, leave it there. Yes, John.
The existing PARP inhibitors have really awful side effects. Thrombocytopenia is 70%, nausea 50%. They're just awful. So what is your plan for dealing with that?
Dr. Lippman's saying that that these drugs have awful side effects. They're not administered alone. They're administered with anti-cancer drugs, toxic anti-cancer drugs. And I've tried to figure out, and unsuccessfully, what are the side effects associated with the drug itself? I can't find that. So you take people who are taking these anti-cancer drugs, and things aren't going well, and they add on, and then they develop thrombocytopenia. I don't know that it's the add-on, number one. But if it were the add-on, it's the add-on to an anti-cancer drug. It's not the inhibitor itself. As far as nausea, we don't see anything in the rats, any sign of writhing or anything like that. They can't vomit, but they can writhe. And you don't see anything like that. Actually, we don't see any particular behavioral effects in those rats. So yes, we don't know what the side effects might be in humans, but that's farther down the road. These drugs are commercially available. Theoretically, somebody could prescribe it. But I don't think we're ready for that yet, for anybody to say, hey, I'm going to try that in a patient. We've got a lot more work to do first. And we will be doing toxicology, initial toxicology, animal toxicology studies, if we can get the SBIR funded.
I thought that was a great talk, Greg, and I applaud you for taking antidepressant research to the next level in terms of addressing anti-inflammatory interventions, which we have a broader interest in the department. But I was wondering, I'm not critical about the use of the PARP inhibitors, but how do you portray them in the larger potential groups of interventions for anti-inflammatory foci and MDD that could be brought to bear, such as inhibitors of pro-inflammatory cytokines like TNF-alpha, IL-1, and IL-6, and PCSK9 inhibitors like evolocumab. Right. Why choose PARP inhibitors specifically against other anti-inflammatory interventions that could be investigated right now for MDD?
The PARP inhibitors are upstream of the cytokines that you're talking about. So we're blocking upstream of all that. So it has a much more broad anti-inflammatory effect. And the truth is, as I mentioned, we don't really know that it's anti-inflammation that's going on. It could be that we're inhibiting chromatin remodeling that occurs during stress. And PARP inhibitors also block long-term potentiation in the hippocampus. So one of the criticisms of the paper, which is now published, somebody was just trying to block us because they said that PARP inhibitors are known to block LTP. So they're going to block memory, and you're going to have all kinds of memory problems. Of course, our paper's about rats, right? So they were just adamantly opposed to us publishing the paper because of the potential. Well, I didn't know that much about LTP and memory, but I knew that ketamine blocks LTP very effectively, and NMDA receptor antagonists do. And it's funny, nobody's talked about memory and ketamine, or not very much. So that was one of my arguments. But then when I went to the literature, I discovered that a lot of antidepressant drugs, SSRIs included, inhibit LTP and memory formation. It's just not to the extent that you have memory deficits, at least as far as we know. I don't know if anybody's really studied that carefully. But many antidepressant drugs block LTP formation. So I argued in the paper that it could be that's how they work. That interfering with memory consolidation from stressful events or to the continued perception of stressful events in one's life may be how these drugs are working. So in anti-inflammatory, we cast that broadly. We use it in our grant because it's such a popular catchword right now in grants and meetings about depression. But that's what we're using it for, as a catch, as a hook. But we don't really know if that's what's going on, and that may not be how they're working. So we have we're very open-minded about how the drugs might.
Saying that it's a problem to block LTP is like saying that an antidepressant has sedative effects. I mean, you know, there could very well be a distinct positive in that. It's not at all clear that that isn't part of the action. I was going to point that out. But the other thing is, what is the route of administration you're using in rats? And how would it be in humans if it were being administered in humans? Is there...
I've not seen the papers, but one of the reviewers adamantly stated, it was the same one that was against it, 3AB is not tolerated in humans. So I don't know what the side effects are. I couldn't find it.
The other thing I was going to say is it seems to me that the criticism about Brodmann's 10 and the uncinate fasciculus is misplaced, because while somebody is saying they want to see the impact in the limbic system, for all practical purposes, the anterior cingulate cortex connects up to Brodmann 10, and then Brodmann 10 connects to the temporal lobe by way of the uncinate fasciculus. So essentially, you've got the tertiary cortical level of the limbic system coordinating both the prefrontal cortex and the temporal lobes together. So to me, that made a lot of sense. I was actually kind of surprised that it happened in the occipital lobe as well, because I would have thought that evolution would have directed it in the one direction, but I suspect it's throughout the brain that these things are active.
Back to your earlier response, Greg, I wonder then if the PARP inhibitors aren't that specific for MDD and might be more generally thought of as treatments for stress burden, and to what extent that might also be true for aging.
And also maybe TBI.
Yeah, well, our patent application is quite broad. It picks up a lot of psychological stress-induced disorders. So we think it might have broad application. And so far we're just studying depressive models, but we have plans, if we get some funding, to broaden that. But that's a very good point.
Just going back to your very early slide, it seems that endogenously the role of PARP is adaptive. Could repeated use of this increase oxidative damage?
I think Jacob, is it you that's going to look at, we're going to look at the treated animals and look at DNA oxidation in the 3AB treated animals. So, because we don't know whether it's going to go up, you know, it kind of, the fact that they were antidepressant, I was kind of surprised, to be honest with you, that they were. I thought it might actually, if I were a betting man with Russ that day, we didn't even bet a beer, I would have bet that they were going to make the animals worse, but it didn't. So I don't know how to predict what it's going to do to DNA oxidation even. Is it going to increase DNA oxidation? You'd think it would. But if that DNA oxidation is a result of the activation of inflammation, for example, and inflammatory mediators coming in and producing more oxidation that ultimately gets it to a significant level, then we might reduce DNA oxidation because we inhibit the inflammatory process. Oh. I can hear you. I would say the data would support that, because we're comparing, in that study, we had a variety of ages. The age range is from like 20 years of age to 70 in that group of subjects. So they're paired with a same age control. So in order for it to be consistently elevated in the depressive, it must be the aging effect plus the depressive effect to make them significantly different. Yeah. Okay, so disclaimer, I'm a pharmacologist, so my view of antidepressant development was heavily focused on pharmacology. Of course, there's psychotherapy, cognitive behavioral therapy, and all that. And I would guess if there were something like that in rodents, it'd probably be very effective at it reducing oxidative load, because you would reduce the stress-induced changes that increase oxidative stress. So, yeah, I'm actually, as a pharmacologist, whenever I hear one of my family members say they may get an antidepressant drug, I spend a lot of time explaining to them why they should think otherwise. So, you know, I'm not an advocate of the use of drugs if behavioral therapy of some kind is equally effective. Yeah. Yeah, decreased levels, right. In rodents or in humans? We haven't looked at any of those. We've looked at antioxidant enzyme gene expression, and it's low in major depression. We published that. I had two arrows pointing down. Yeah, that's all humans. On one column was humans, the other column, the one with very few things, was rats. Yeah. Yeah, and people tried to treat depression with antioxidants. We all know that story. You know, the data's kind of not great. Okay. Any other questions? Okay, thank you.